Oh, you've already got them teed up. Great. So to build off of Lynn's introduction, this case study, actually, when we discussed it in the organizing committee, one of the aspects to it is, as a case study, is it brings in alternative approaches, both in the original Tier 1, Tier 2 approach with in vitro methods, but we're also going to talk about how high throughput and computational approaches can be incorporated into our testing and screening battery. As was mentioned earlier, this framework is statutory. We have a science-based, risk-based decision-making process. One other context, and it was alluded to earlier, but I'm going to underscore it, is no decision is a decision. So the absence of data leads to no decision, and that no decision means that there's no risk. And in the context of endocrine disruption or developmental reproductive toxicity or developmental neurotoxicity, the endocrine system is actually critically important on the organizational events that structure and function follow. So it's important that the endocrine system during development functions normally. The other concepts that are going to be illustrated by this particular talk, and it's not really, I'm not going to be able to address trust so much, but I think what you'll hear from me in this case study is the issues of confidence and uncertainties, and how, at least as a risk assessor, I'll put my risk assessor hat on, how we're trying to address confidence and uncertainty in this new testing paradigm. And again, for confidence, we're talking about assumptions in the data, and then for and transparencies. And then some of this is qualitative, some of it's quantitative. So let me go on and start the talk. So my typical disclaimer, again, as an agency representative, I'm not representing the agency in any of my offhanded comments or responses to questions, but what you'll see in my presentation is basically, it is cleared by the agency. Let's see if I can get forward. Okay, so as Lynn alluded to, we're not, it's in a static mode. And in 1995, we actually put out a Federal Register notice announcing that we were considering alternative approaches and a high throughput approach, a computational approach for alternatives to the current Tier 1 endocrine battery tests. And what I'm going to talk specifically about is the uterotrophic, the in vitro tests and the uterotrophic assay for estrogen, as an illustration for some of the work that we're going to do for the other endocrine pathways. So we took comments on this. We received quite robust comments from limited sources, stakeholder communities, including, per example, NRDC, EDF, the ACC, you know, these different abbreviations as different stakeholder groups. And many were, and the Humane Society and PETA as well. So some were very much in favor for us taking these approaches and moving forward rapidly. And others were saying, go slow, we need more validation, more evaluation. So what have we done? So the EDSP's goals for using these computational approaches, and these really focus on rapidly screening chemicals for endocrine bioactivity. As Lynn pointed out, so we started out with a list 1 of 62 chemicals. We quickly realized, taking 3 to 4 years just to get list 1 data and evaluate list 1 data, that we needed to have a faster, cheaper, more efficient process for getting through the large number of pesticidal ingredients. And pesticides, the statute says pesticidal products. It doesn't say pesticide actives only. It doesn't say just the pesticide uses. It says pesticides. So that's the inactive, the inerts and the other portions of the formulations are supposed to be assessed too. And it's both new pesticides as well as existing pesticides. Right now we have focused on the existing pesticides. So again, a framework for rapidly assessing the pesticidal actives as well as the inerts and potentially other industrial chemicals, both with the SIDWA context, safe drinking water context as well. The screen would contribute to the weight of evidence in determining the chemical's potential bioactivity. Again, we're not saying disruption with tier 1, we're talking about bioactivity. And the focus is on a low false positive rate, but still preserving, having a very limited false negatives. So we're trying to hit that sweet spot between false positives and false negatives. And then provide alternative data for the current EDSP Tier 1, particularly the in vivo assays, has also been a focus, and to screen thousands of chemicals. I'll repeat myself. The ultimate goal is to take this from the estrogen pathway to the androgen pathway and onto the thyroid sterogenesis broader context for what we've defined. Now, I would say next week we're having a peer review of our androgen work and our sterogenesis work as well. So that's at our SAP next week in Potomac Yards. Stay tuned, and I think we've moved quite far along with our androgen model as well, similar to what you'll see presented here today. So for the Tier 1 assays, essentially our results today are, we believe we've done the work to show, I'm going to tell you what I'm going to tell you, we've done the work to evaluate and validate the estrogen computational model and high throughput assays to, as alternatives for the estrogen receptor binding assay, a low throughput assay, and the estrogen receptor transactivation assay and the Uterotrophic assay. Those are all lower throughput assays that we've either replaced with a high throughput assay or assays. And again, I'm going to talk a little bit about the approach as we go further. The others are listed here, are work in progress and sort of targets for when those particular computational approaches and batteries will come into fruition in the future. Again, assuming continued funding for the EDSP program. No political statement there. EDSP's program has shifted away from the apical endpoints per se and focusing on the toxicity tests in rodents to more of human-based pathway approach and disease-based approaches. And so adverse outcome pathways has been the focus more so in the more recent developments of our screening approach. And we still are statutorily required to get back to measurement of prediction of in-life dose response. So we're screening and prioritizing, and then we go to that. So the exposure is still relevant, and I'm not going to talk about exposure today and the high throughput exposure approaches, but that is part of the program. And we're, of course, incorporating monitoring data and real-world predictions into that exposure context. So our recommendations here are, our approach here is consistent with the recommendations that we've had from previous science advisory panels in EDSTAC as well, to use computational approaches for screening and prioritization, which include QSARs, quantitative structure activity relationships, and other high throughput approaches for toxicity testing as well as the exposure, and the AOP approach. So the framework here that I want to describe has two legs. One leg is actually the internal leg is actually developing high throughput and computational models and validating those by testing hundreds to potentially thousands of chemicals and developing the data for variability analysis and uncertainty analysis around those different assays and on the predictions of those assays. In addition, in parallel to that, one of the key parts of the evaluation validation aspects of this is not throwing out the previous in vivo data or the previous tier 1 low throughput data, but using that data as a benchmark. And so we have that data as the reference data, and we have positives and we have negatives from that data. those in vivo assays and those previous in vitro assays, as well as literature review. And what I say by literature review, and this comes back to my confidence building aspects of this developmental process, is really developing systematic review protocols. And my slides, which are actually available as a handout, gives you some of the protocols and some of the specific criteria of those systematic review protocols are critical for transparency and understanding and recapitulation of why we chose a chemical to be a reference chemical. So we've chosen these reference chemicals, both positives and negatives, not screaming hot positives, pharmacological agents, but agents with a range of potencies, which comes up in my next slide. So that's a critical aspect to the testing procedure and the validation procedure, and gets us at model performance and the validation in chemical screening. So reference chemicals include a range of structures, a range of potencies to accurately to detect an effect and predict effects. And what I'm going to speak to in subsequent slides is that prediction, predictive validity. And the in vitro and in vivo reference chemicals, again, from the systematic review, we were able to obtain not a dozen of chemicals, as many times or less than a dozen is what's usually used in an OECD validation protocol, but more than several dozen chemicals. And the other question that we had, and again, this comes from the visionary thinking of folks in our Office of Research and Development, as well as our assistant administrator at the time, Jim Jones, who was very much looking to how can we use these approaches in our decision-making process? How can we be more efficient, more effective with the resources that we have, recognizing that we've got all these pesticides and inert ingredients, and we don't have enough animals and capacity, lab capacity to test all these and try to meet our statutory deadlines, which we were behind on. So our evaluation and validation process, again, are not necessarily new, but peer review was a key part of our evaluation process. Again, getting the buy-in, involving the public, involving peers and experts in the field of endocrinology, in clinical endocrinology, in epidemiology, in the review of our approach. And we've gotten, at times, some relatively critical input in that process. The public engagement has also been a key part. So two websites web links are listed here. One is to the Science Advisory Panel. You can see we keep a record of all of our reports, as well as our response to comments to those recommendations. As well as our docket contains all the data, all the supplementary files. We're constantly filling that docket with additional information about the validation process. I list in the next bullet here the OECD validation and international acceptance criteria or considerations for updated test methods. Again, this is the OECD Guideline 34. That's the core of what we use in our evaluation process. There are some exceptions, and again, one of the things that we're shifting to is moving away from what had been done with the EDC program with ring trials and multi-lab investigations and validation to performance-based testing. And developing performance-based test guidelines. That's the new realm that we're entering into, and Dr. Lowett will talk more about that in subsequent talk here in later session. The other key aspects of validation is really addressing the characteristics or addressing reliability, relevance of the methods for specific purposes, again, a fit for purpose context, and then relevance and reliability of our high throughput approaches for both the in vitro low throughput and the the in vivo uterotrophic assay. So I'm running out of time. Again, quickly here, most of the concern about environmental estrogen estrogenic compounds is in the agonist realm, less so in the antagonist realm, which is unlike the androgen pathway. But again, we have tests for both agonist activity as well as antagonist activity in the battery. And the tests that we're looking at for alternatives are the ones that are circled here, the 3 that are circled here. A little bit of background about the Uterotrophic assay. It is a short-term in vivo test. It's an overactualized animal. It's a female rat, generally a pubertal or adult rat, not always. And what you're doing is you're adding an agonist as a positive control, estradiol, and you're looking to either the antagonistic activity of the compound or the agonist activity of the compound at several doses. So you're looking for, as a bioassay, really an increase, generally, an increase in the weight of the uterine horns. Because estrogen, the response to estrogen is an increase in weight. So that's the basis of the in vivo assay. We've done systematic literature review, identified through PubMed and other sources, other databases with FDA and other agencies. Again, chemicals that have been tested through uterotrophic assays, guideline-like studies. We've looked at these, sifted these based upon the criteria that I've listed in the addended slides. We've come up with quite a large number of papers, quite a large number of descriptors. It takes 2 reviewers to systematically go through all this. It takes an expert, semi-trained expert to do these kinds of systematic reviews. It's not a novice that does this. And then we come up with a number of chemicals that are positive, that have data, reliable data from this systematic review process. So those systematic review provides the basis for the one leg of that validation process, which is for our external validation of the assays and the model, computational model results. So I'm going to jump to due to time, jump to the bottom line here. The true positives, we had true positives of 28 chemicals identified as true positives. We had true negatives, 12 were identified as true negatives from our systematic review and testing. We had 1 false positive and 1 false negative. And again, I'll explain partly the reason for false positives is not testing at a high enough concentration in the in vitro assays. And false negative, again, has come up earlier. At least the reason here for this particular one was lack of competence, metabolic competence in the assays. Again, can provide an opening for in the screening process for chemicals to get through the screening battery. Our overall accuracy was 95% in our predictive validity. Our sensitivity was 97, and specificity was 92%. So we really, at least in comparison to the in vivo assay, we're doing as well or better than the in vivo assay and the in vitro assay, low throughput in vitro assays that we had currently in the tier. So our alternative is working, as well as the tox testing assays in the current battery, or better. And these, of course, are the publication links, series of publications. Each of these publications have supplementary data files. An important aspect to the overall transparency issue was putting that all that supplemental data out to the public so the public could actually see that data, and members of the public and stakeholders whose particular chemicals were tested came back and also did additional analyses with the same data. So this particular slide, I know it looks busy, but the main point I want to try to emphasize here, and I don't have a pointer, but the main point is if you look at the molecular interactions box, the upper part of the table really emphasized where taking the adverse outcome pathway approach, looking at the estrogen signaling pathway in a more complete way rather than just the transactivation and the estrogen receptor binding. We have more biological coverage. So that's a key aspect to the high throughput computational approach that we're presenting and pursuing on, is covering more of the biological space, having more assays that are orthogonal to the pathway, and have different types of detection systems. So our strength of our computational model is beyond the limits of one particular assay, a receptor binding assay, positive or negative, or concentration dependence of that one assay. So that's a key aspect to this battery approach. So in summary, our ER agonist, this is the area under the curve model, developed using the high throughput data, and right now it's currently 18 assays in the battery. We're evaluating additional aspects of those data, looking back to say, what's the fewest number of assays that'll give us the same predictive validity? Do we need all 18 assays to get the same predictive validity? And the answer is in a submitted publication that'll be coming out in the very near future. The short answer is we don't need 18 assays. And the other aspect to that investigation is really coming up with what are some of the critical performance criteria, performance considerations that we want to include in a performance-based guideline, so that if a stakeholder or academic or others were testing, what kind of information would you have to complete and document and annotate for your assays in order for the agency to accept it. And one of the lessons learned, and I'm jumping ahead, was annotation was critically important for acceptance. There appears, at least from our work with the high throughput work, both for ER and AR, was that the in vitro AUC values in the computational model has a relationship to the activity, the bioactivity in the current EDSP screening assays. And the results indicate that at least the ToxCast data, which is the ToxCast, I use that term, it's Tox21 data and ToxCast data, broadly does provide rapid and effective resources and approach for identifying substances for potential ER bioactivity. Again, potential. And limitations, I've touched on the limitations briefly. Others will talk more about this, and this is general to alternative approaches. The first one is metabolic competence, metabolism. Do you have a metabolically competent system? And the other, again, is solubility issues. Again, some of the reasons that we have false negatives is we're not able to, because of solubility, not able to test this at higher concentrations, and where positives have been identified. They've been identified as positives at high doses. And some of these are actually pharmacological agents. They're used in pharmacological contexts at high doses. We're not necessarily able to test those in vitro in small volumes and in DMSO. So DMSO being that's the main solubilizer that we use. We're trying to address both the metabolism issue with new technologies. We'll be addressing the DMSO issue with additional technologies and water-based systems. And then there's several other classes of chemicals. Reactive compounds don't work very well. Metals don't necessarily work very well. Particulates don't work well in our our low volume, high throughput assays. And then we have volatiles. And volatiles, we've also got a new technology, a new approach for testing volatiles. So there's a lot of research and development that's ongoing here on these alternative platforms that will be relevant to both the EDSP program, but also to the subsequent conversations about high throughput testing alternative approaches for toxics and others. So conclusions, I want to wrap up here, and this will sort of lead into our discussion. Transparency has been one of the critical issues that we've gotten a lot of comments about, both from our science advisory panels as well as the public in the public comment periods and in our Federal Register notices. Lessons learned about not having all your data out there in the public for others to analyze, something that I think the ToxCast platform and the initiative has been very forthcoming about, but again, annotation of the assays, a recent development on our part is to annotate, again, according to OECD guidelines, all the aspects of the particular assays. And again, we've provided input back in our leaders in the OECD validation efforts on how to better annotate in vitro alternative assays. Models, again, providing model code for anybody to use, open source model code. Again, you'd be surprised how many in the risk community use models that are not publicly available and present those to the agency, and then we have to peer review them and say, okay, well that's difficult. How do we get this to the science advisory panel members who don't have that particular proprietary model? Or how does the public evaluate or have confidence in this if they don't have access or can't get access to this model? And by public, I include academics as being part of that. Peer-reviewed publications are also critically important, and providing supplemental data also critically important. I didn't mean to skip over making it transparent what your evaluation process is. And again, we have taken in numerous occasions, and we'll be doing this as well next week, what our evaluation process, what our direction is for evaluation to our science advisory panel, taking public comment on that as well. And that also provides lessons learned. It provides transparency to the community. We hope to build confidence in that process. We have built confidence in the process, and we've learned some important lessons from some of the public comments about lack of transparency in the evaluation process through those mechanisms. We're, of course, working towards using more web-based tools, and our endocrine disruptor screening program dashboard is currently frozen in time. We'll be working with a more real-time, I should say, update to our dashboard, have, again, tools that are more interrogative and user-friendly for not only risk assessors inside the agency, evaluators inside the agency, but folks in the external community to also evaluate the data and get access to the data that we have and data that's we've also pulled in from our systematic reviews. Public engagement, again, I've mentioned public engagement as part of our confidence building, our stakeholder engagement. Both of these are critical and again, get at not necessarily trust per se, but confidence building and being able to say where the uncertainties are, describe the uncertainties either in our models or in our predictions in a qualitative sense, what the limitations are, and in many cases what we have done is provide quantitative estimates, probability estimates for the data, for the assays, based upon the chemical space that's covered and the biological space that's covered. So that's been an ongoing endeavor, and again, could not be accomplished without a team effort. And then the other thing with regard to the endocrine disruptor screening program in particular has been very engaged with stakeholders on data evaluation, standardized templates, standard evaluation protocols. Again, from public comments, one of the things stakeholders want to know is how are you going to evaluate this data? How are you going to evaluate a positive and negative? How's this going to work? And we have put more effort into that than many other programs, testing programs, screening programs throughout the world. And in fact, ours are being used as a model by others. Peer review, again, the peer review was quite positive for our estrogen receptor model, and the links are provided. As I indicated, this was a team effort. It really took partnership, a multi-agency partnership, multi-office partnership within the EPA. Includes my office, that I'm the acting director of the Office of Science Coordination Policy, the Office of Pesticide Programs, which actually manages the test orders for the Tier 1 tests, and collaboration with the OPPT, Office of Pollution Prevention and Toxics. Our Office of Research and Development really led many of the research efforts, as well as NIEHS. We also had many consultations with our Office of Water, again, because of the SIDWA context to the chemicals and the identification of those potential human exposures that would be part of the pre-screening for inclusion in the list making. Our National Center for Advancing Translational Science, NCATS, again, in Maryland, was critically important in much of the screening assays, the robotic screening assays, and then NCATS is a critical partner in our evaluation of alternative approaches and the validation with ICVAM. So all of these partners together really helped us, not just with the ER computational model validation, but are continuing to be partners with our additional efforts on androgens, thyroid, and the steroidogenesis pathways. Thank you.